My name is Scott Osborne. I'm the Vice President of Economic and Corporate Engagement at The Ohio State University. Good afternoon, everyone, and thank you for joining us. Welcome to this showcase, today's showcase. I lead the Corporate Engagement Office, which includes technology commercialization and the Keenan Center for Entrepreneurship, where we help spin out Ohio State startups and mentor entrepreneurs, among other things. The Corporate Engagement Office is very pleased to be co-presenting the COVID-19 Startup Inventor Showcase with our friends and colleagues in CCTS and the Nationwide Children's Hospital Office of Technology Commercialization. Those of us who work in the commercialization realm on a day-to-day basis realize how many inventors and potential inventors don't fully understand how to move your ideas forward into the marketplace. I'm confident that today's program will educate and inspire you, and I want to thank CCTS for taking the lead on organizing this. Now it's my pleasure to introduce Jennifer Dower, who will introduce our speakers. Jennifer is the Chief Strategy and Transformation Officer and Vice President at The Ohio State University Wexner Medical Center. She started at the beginning of July. Prior to that, she was Managing Director at Cincy Tech. Prior to that, she was Senior Vice President for Strategy and Growth at Cincinnati Children's Hospital Medical Center. She spent the bulk of her career at P&G, her last post as Vice President and General Manager for New Business Creation, Global Beauty and Grooming. Welcome, Jennifer.
Thank you, Scott. And I'm very pleased to be here. Hopefully everybody can hear me. You know, as Scott said, I think the showcase like this, a forum like this, is a wonderful opportunity to give people exposure to and visibility on what's going on in the system, how are others approaching this and tackling this. And I can tell you, whether it was my experiences at Cincy Tech, for those of you that don't know what that is, it is a venture capital firm. We invest in early stage startups, or they invest in early stage startups, including a couple out of Ohio State, many out of Cincinnati Children's and other academic institutions. And having the opportunity to partner with people to see those innovations come to life is extremely, extremely rewarding, wonderful for the beneficiaries, whether those are the investigators, the researchers, the communities that benefit from those transformations, or in certain instances, the investors. And so without further ado, I want to introduce our first speaker, Dr. Carlos Castro, who is the principal investigator and the faculty lead of the team from the College of Engineering, who will take you through their experiences in this space. Carlos?
Great. Yeah, thanks, Jennifer, for the introduction. I'll tell you a little bit more about myself in a minute and the team, but I just wanted to start by thanking CCTS and all the organizers of today's event for the opportunity to be here with you and to share our story with you. So we are a team of researchers, which, as you see on the slide, is the nanoengineering and biodesign lab side of our story. And we're also relatively new inventors, which is the dynamic nanorobotic assays and technology side of the story, which you see on the right, which we call DNA Tech for short. DNA Tech is our concept for a startup, which for now is really just a concept, but that's the path we're working towards. And we're excited to be able to partner with a lot of great people, both at OSU and outside OSU, that you'll hear a little bit more about later. But I'm going to focus on telling you more about our research story, which is, of course, where our path as inventors really began. So as we started the lab, you know, just almost 10 years ago now, the mission that we started out with was really to use DNA devices to understand the physical properties and mechanical design of biological systems, spanning from single molecules all the way up to animal model systems. These are some examples of the experiments that we've done in the lab. And as you see on the left, the devices we build are actually on the same scale of the coronavirus itself. So that gives us a nice opportunity to build devices that actually interface, you know, either with the virus or maybe the materials that make up the virus, which is, again, kind of what we're focusing on today. But we've also implemented these devices across a range of molecular, cellular, and all the way up to animal model systems, as you see in the images there. That research mission has really branched out into a lot of different directions, including things that I never really thought I'd be doing, such as thinking about a startup company, which brings us to DNA Tech, which we ultimately envision will be able to solve problems in health and medicine using cutting edge biotechnology to hopefully make the world better. And that's really been quite exciting over the last few months for us to get a chance to think about directly translating our research foundation into making an impact on societal problems. In particular, of course, if we can make an impact on the current pandemic that the world is facing. So before I tell you a little bit more about our research, I'll tell you more about myself. I am a Buckeye through and through. I got my bachelor's and master's degrees both in mechanical engineering at Ohio State University. I went on to get my PhD also in mechanical engineering at MIT. And then before coming back to OSU, I was a postdoctoral fellow at the Technical University in Munich for about a year and a half, and came back to Ohio State in 2011 to join the faculty here. A couple of personal details that are, of course, also important part of my life. I was born in El Salvador. I grew up in Ohio for the most part. Being Latin American, soccer ran in my blood, and my dad made sure of that when I was a kid. So I actually also ended up playing soccer for Ohio State as an undergraduate student from 2000 to 2002. And then fast forward about 20 years to a few months ago, the beginning of this work that you'll hear about today was entwined with homeschooling a first grader and a preschooler, as well as having a new baby at home. So they've kind of inevitably been a part of this invent from home journey. And of course, my wife has also played a big support role in helping out at home. And so that brings us to our laboratory. You can see a picture of our lab. This picture is a little bit out of date because of course we haven't been able to get together as a group for the last several months. But you can see the makeup of the team. You can read about the makeup of our team on the top here. I won't say too much, but really the main thing I wanted to point out is that we're a highly diverse group, both in terms of background and training. So actually our group members come from 10 different countries, as you can see on the list below. And from a training background, we represent, mechanical, biomedical, chemical engineers, biophysicists, biochemists, biologists, right? And that's just even all members of the lab. And of course, we collaborate very actively across campus and outside OSU with a wide range of groups, which I think has been a big part of our success. So with this group of talented people, what we actually do is to design and build DNA devices for a range of applications in nanorobotics, medicine, and biophysics and bioengineering. You can get a sense of our design capabilities at the nanoscale with the image of the Script Ohio there. That was actually a side project that my first PhD student worked on, is about 100 nanometer scale Script Ohio, which actually got him into the Guinness Book of World Records. But we have pretty unique and advanced design capabilities at this scale. And then the next few slides, I'll show you how we've been translating that into applications over the last several years. So what I really consider the core of the lab and where we have, I think, truly unique expertise is in the development of these dynamic nano devices. Along with a collaborator in my department, Professor Haijun Su, our lab has pioneered the development of these DNA nanomachines that look, move, and function similar to macroscopic machines. You can see several examples here. For example, on the top left, a joint that is much like a hinge that has rotational motion or sliding motion in the top middle there. Or the other examples show different kinds of complex 2D and 3D reconfigurable mechanisms. And we've also just recently started working on computational modeling to get a better quantitative sense of guiding the design of targeted functions, such as things like conformational changes, which again is important for the sensor you'll hear about a little bit later. That you see a video of that computational modeling on the bottom right there. And then in the last several years, we've been taking this design experience and dynamic devices and translating those into biomedical applications. Again, these are a couple examples. On the left, you see an example showing we can install these DNA devices actually onto a cell membrane, which those are the fluorescent rings that you see on the images on the left. And the idea there is to use those devices to probe or control cell-cell adhesion, or even detect biomolecules at the cell surface, hopefully in a native cell environment. And on the right, you see a highlight of some of our targeted drug delivery work, where we're collaborating with researchers in the OSU Leukemia Research Group, including John Bird and Carolyn Larkin and others. And that red signal you see there on the video on the right is actually DNA structures that have been taken up into, in this case, a leukemia cell line model. And then lastly, we have a broad range of other applications. Here we're just showing some highlights. On the left, you see magnetically actuated DNA devices. There on the video on the bottom, hopefully you're able to see we can actually open and close nanoscale hinges in real time. So that gives us a nice avenue towards robotic applications. We can work on patterning and reconfiguring gold nanoparticles or other biomolecules, as you see in the middle. And on the right, you see this is another active area of research, is using devices to probe and study the structure and conformational dynamics of molecular complexes. That example actually shows a nucleosome, which is the fundamental packing unit of DNA that makes up chromosomes. And that's a big set of work that we've done collaborating with folks in our physics department here. And so that hopefully that gives you a nice overview of the different activities and the capabilities that we have in the lab. And I'm going to hand things off to our research engineer, Patrick Halley, to tell you more about how our team has been translating this research foundation to develop rapid diagnostics for COVID detection.
Thank you, Carlos. Let me start by saying I don't know how many other labs have the amount of open communication, trust in their staff and students, and collaboration that this story has. So thanks again for providing such a space. My name is Patrick Halley. I'm a research engineer in Carlos's lab. Overall, I'm an engineer who dabbles in biology and not the other way around. Though I know enough about enough of biology that I specialize in research that combines engineering design with biomedical and clinical applications. Today, I'm going to start talking about how we went from idea to invention, and we'll start with where most innovation starts, where it's the problem. What is that problem? So it's COVID-19 diagnostics. Obviously, you know that, or you wouldn't be here. And I'm not going to go into the numbers too deep because we've all been living it for the last 5+ months. But since the pandemic began, COVID-19 detection has had many difficulties, and many of those that we saw in March are still current problems today. And on the flip side, with so many great scientists around the world that are working on this, it's a quickly changing field, and though we try to keep up with it, you'll have to forgive me if I miss something. Reverse transcriptase polymer chain reaction is a highly accurate test and is still considered the gold standard, but requires a good amount of time to perform. Many of the supply chains are still having backlogs, and the collection method is very intrusive. And to let you in on that secret, avoiding them poking my brain with the giant Q-tip is pretty much a big motivation factor for me. And luckily, just this Saturday, a saliva-based PCR assay was approved by the FDA, which will hopefully help solve that problem soon. The other method that has had FDA approval since early on is antibody or serological testing. It is a blood test that's rapid and indicates the presence of anti-COVID antibodies. This likely means that once you get an infection, or that you once had an infection, but does not mean current infection, nor does everyone who gets COVID-19 have them. Now, some of the more emerging tests that are out there can be broken down to a few different groups. Isothermal, which is or known as single temperature reactions. There's CRISPR and antigen testing. Now, antigen testing is the one that was in the news recently for the false positive test of our governor. They each have a potential to improve diagnosis of COVID-19, but of course, each have their weaknesses. Now, because of time, I'm going to actually focus on the gold standard. And if we look at the workflow process, it contains 5 steps and then a readout. RT-PCR works by breaking open the viral envelope, purifying the RNA, converting that to DNA, and then amplifying the DNA. Many of those steps are time-consuming or labor-intensive. Our solution is to stick with this general workflow with the option to use the same equipment for our readouts, but to eliminate some of the more time-consuming steps. How do we eliminate those steps? By amplifying the signal instead of the DNA. Now here's a representative image of our DNA origami nanostructure, which is a type of dynamic nanostructure or hinge. It's roughly the same size as the virus, as Carlos said, and if we take a closer look at it, it has many fluorophore and quencher pairs acting as one. Those are represented by the green and black balls on the middle of the hinge. So those are going to actually align when the hinge is closed, so the signal turns off. But when it's opened, when it's triggered, it will shine very, very bright. And that means we need only a limited amount of hinges that open to have a measurable signal. And I'd love to share more about the design and its interaction with the virus. But we're actually still in the process of filing our provisional patent. And this is actually not intended to be our public disclosure of the technology. So instead, I'm going to keep talking about the process of ideation. And I know nobody wants to do this, but we're going to go back to March. It's March 16th, and the lab and campus have quickly closed down. I was in the mountains myself on vacation and didn't get back to work till Wednesday. A few a few work days home later, and I'm officially going stir crazy. And you might not be able to tell, but I don't slow down well. At this point, I officially want to do something about it. But like most people, I'm feeling pretty helpless. I tossed some ideas or some thoughts around, and about a week later, some of those ideas or those thoughts become turned into an idea. And I take a dive into literature to see what I can learn about the virus and its diagnosis. About a week later, I've drawn up some items, and I've sent them to my research partner and good friend, Dr. Chris. He responds very quickly, and luckily, Chris is a biologist who dabbles in engineering instead of the other way around. And now that Chris and I have sorted out like the basic feasibility of the process, it's time to bring it to Dr. Castro. And you might wonder why we didn't just go to Carlos from the beginning. Well, he was pretty busy himself. As you can see, he's also got a problem with idleness. A collaborator at the Comprehensive Cancer Center approached him about whether the mechanical engineering department could 3D print some face shields. Well, we could. And after changing the injection to a injection molding process to scale up, using a collaboration with the Integrated Systems Engineering Department and some members of the ME machine shop, engineering fabricated thousands of face shields for the CCC. But we're going to go back to the diagnosis story now. And so we have brought this technology to Carlos, and he likes the idea, but he has some suggestions. They are, of course, good ones, including how we could control the energy landscape of the opening and closing of our hinge. He also suggests bringing in Melik Agha, a leading grad student in our lab, which was, and of course is, a great idea. She previously worked with Biosciences and was working with a structure that we could adapt for a prototype of our design. Over the next 4 days, we quickly write up an application for the Office of Research COVID-19 seed funds, only to miss the deadline by about an hour, which just meant that the decision would not be coming for a couple weeks because they had a 3-week open window. In the meantime, we had a brainstorming session with the 4 of us, so we continued to talk and collaborate. We came up with some other ideas or other items that could make our original idea more useful, and including ways that we can improve it, including contingencies that were just in case it didn't have low enough sensitivity. On April 25th, we received notice that we thankfully received one of the seed grants, and here's where we really hit the ground running. We quickly realized that if we wanted to get this into the world and to actually make an impact, we needed to protect the idea and make sure no one else was working on something similar and we weren't stepping on any toes. Before we could do that, we needed to talk to Alex. Alex Maris is currently a postdoc researcher at the University of Chicago and a former member of NBL. He was one of the lead people in the development of DNA nanostructure, or dynamic DNA nanostructures. He is also the person who created the original design of that hinge that we adapted for our prototype. Essentially, if Alex had never been a member of the lab, this invention never happens. So we Zoomed with him and we talked through some ideas, and he had some few of his own, of course, and we included him as an additional inventor. The next day, we contacted Janelle of the Corporate Engagement Office, and she encouraged us to file an invention disclosure. So of course we did. And here's where our story really splits. And we can no longer talk about it on this timeline. So it basically, the project split into 2 paths. They are both very reliant on each other, but we had to, but it's easier to focus on in 2 points. So those are research and commercialization. I'm going to pass it off to Melik Agha, who is our PhD candidate, and she's going to walk you through some of the validation of our technology.
Thank you, Patrick. Hi, everyone. My name is Melik Agha, and I'm a 4th-year PhD candidate in mechanical engineering. I'm originally from Iran, and I came to Ohio State in the fall 2016 and joined Nanoengineering by Design Lab. I'm also a 2020 graduate Plutonia Fellow, and my primary research is focused on biological applications of DNA origami and to use these nanostructures to explore biomolecular interactions of cell membrane with biomarkers in extracellular matrix. I will go through our product and preliminary data we have so far to validate our structure and its functionality. DNA origami as our technology offers advantages which are very beneficial for development of a biosensor. We will have fast and scalable fabrication with total cost of 18 cents per milligram. Tailoring the platform toward a new target such as flu would need very minimal design changes, and the structures are all nucleus resistant. We designed our DNA origami structure using DNA technology to provide greater signal per copy, which results in higher resolution in the output. We would need no RNA purification. We are looking at saliva-based collection at the end. It won't be limited to COVID-19. There would be a 2 simple step process and would be fast, accurate, and versatile. If you look at, next I'm going to talk about our design. Our nanostructure, as you can see on the left, has 2 open and closed states. As Patrick says, when the structure is open, the fluorescence signal is high, as you can see with the blue line on the right, And then when the structure is closed, the signal drops, and as you can see with the orange line on the right. So you can see we have a minimum and maximum signal, and the general idea is the structures are all in the closest state initially, and they will open up upon the addition of the SARS target sequence. But the secret sauce is actually the closing and opening mechanism, and how we are especially developing those mechanisms for this project. Next, you can see some team images showing the structures in open and closed states. And next, you're looking at one detection experiment with it. So the structure in the closed states, the signal was very low, and we added the SARS target sequence, and the structure opened and the signal went up, as you can see in the blue line. And structures, as I said, are all modified with fluorophore quenchers, and we initially started to use FAM and black hole quencher. But we realized we can use more and more fluorescence quenchers. So we went from one fluorophore quencher pair to 6 fluorophores, and realized we can have much higher output signal. We also explored using LXO488 instead of FAM, and realized 2 LXO488 fluorophore has a signal compared to 4 FAM. So that's also an option we can explore in future to use LXO488 to get a better output signal. As we are still in a validation process, but we have some estimation to compare our platform with real-time PCR, which is a gold standard currently for detection of the COVID. We are looking at 45 fluorophore per target RNA compared to 1 that real-time PCR offers. We are looking at 45 minutes total time compared to 6 hours of the PCR. We are looking at 95% accuracy in the saliva collection, much higher sensitivity, the same specificity, and we are, as I said, looking at saliva and nasal swab. As we are developing our product, we are exploring the addition of other options to our structure as well. The first one is to add a cascading reaction where when one hinge opens, it will open up many other hinges. This results in the application of the output signal at much higher resolution, and it can amplify the output signal we'll get in future. The next one is to develop a color changing diagnostic platform for point of care, which allows for naked eye reading. This design could still multiplex with other set tests such as flu. It will be cheap, and it could be repeatedly used everywhere, such as work, school, sporting events, or even in underserved countries. Now we'll hand over to Dr. Christopher Lucas, my colleague, our research scientist, and he will talk about our commercialization journey in the past couple of months.
Thank you very much for the introduction, Melika. Hello, everyone. I'm Chris Lucas, a research scientist working with Drs. Carlos Castro and John Bird to develop DNA-based nanodevices for biomedical applications, including therapeutics and now a novel biosensor to detect the coronavirus. When I'm not in the laboratory, I just a little bit about me. I love to be in the outdoors with my family. And I'm very excited to teach our 2.5-year-old son all about science, the great outdoors, and Ohio State football, hopefully in the spring. So today, I'm very happy to share our story thus far towards commercializing our technology, a true team effort where all members, our partners, come together. We like to think of this as coming together around a table, around an innovation, which is our diagnostic biosensor in the center. My role in the project has been to serve as a liaison between our technical team and our commercialization partners. So as Patrick mentioned, here we have our technical team surrounding the table. Early on, received, of course, as Patrick mentioned, the Office of Research COVID-19 seed grant, which Dr. Jan Weisenberger, who follows me up here, will talk about the role of the Office of Research and their support. As Patrick also mentioned, we quickly reached out to Dr. Janelle Ezel, our licensing manager at the OSU Corporate Engagement Office, to introduce her to our new idea and asked her whether or not it warranted an invention disclosure. And of course, Janelle, we worked with her for several years previously on her nanotherapeutic avenue with intellectual property filings, and she said, go for it. So we went ahead and filed an invention disclosure. Yeah, that was in April. Now, as Patrick mentioned also, Patrick and Carlos together, we decided to reach out to Dr. Art Gouray as well with the Corporate Engagement Office to discuss the commercialization potential of the technology. And Art was also very supportive of our efforts and strongly recommended that we submit a Partnerships for Innovation grant proposal sponsored by the National Science Foundation, which would support proof of concept product validation research. So We just submitted that about a week and a half ago, so I want to thank all of our partners for helping us through that process, especially with the commercialization, which is all very, very new to us. So around the same time, Janelle also connected us with Dr. Kate Schulhaus and New Ventures at the Corporate Engagement Office when Patrick found an advertisement about an upcoming event sponsored by Revone Ventures, a local startup company support studio, and you'll hear from Moo Yang from Revone here in a little while also. The event was a 3-day customer learning lab that would cover core business concepts, as well as introduction to market survey research and minimal viable product, to name a few concepts, all critical for new entrepreneurs. And Kate thought it would be a great experience for us as scientists and engineers to attend that. And she she introduced us to Moo Yang and Matt Chimes over at Revone Ventures. And the next week, or the very next week, we had an idea concept call with Moo and Matt. They also encouraged us, or they were excited about our ideas. They were very supportive and also encouraged us to attend the customer learning lab, which we did. And I think I speak on behalf of Patrick, Carlos, and Melik Agha, all technically minded scientists and engineers, that the customer learning lab experience was absolutely fundamental for entrepreneurial development and growth. During that time, over the course of 1 week, we performed market survey research through interviews of key opinion leaders, including clinical lab directors and end users, both at the Ohio State University Medical Center as well as Ohio Health, to validate our problem, our approach, as well as our product and the idea concept. And it was through these interviews that we connected with Dr. Jean-Michel Balada-Lassatte, who is supporting our efforts to test our device in clinical samples at OSU, and also Dr. Nicholas Wangschwart at Ohio Health, who is serving as an outside mentor. We also met during this time with CCTS's Carolyn Crucifoli and Michael Camp, who I had met last fall in an entrepreneurial training program sponsored by the CCTS. And they also were very excited about the platform and the ideas, and offered some great next steps for us to take towards commercialization. So thank you. We also then very recently through the CCTS, we met with Robert Rengel, who is helping us facilitate the discussion towards regulatory efforts towards filing emergency use authorization with the FDA. So at the conclusion of the customer learning lab, after giving our idea concept pitch for our device, we had a deep dive meeting with Moo Yang and Wayne Embry with Revone to discuss the potential ways to develop our COVID-19 biosensor moving forward. And at the conclusion, we were very fortunate that afterwards Revone offered us an engagement opportunity as new clients to help us drive our technology down the pathway to commercialization. Now, lastly, I wanted to mention that our corporate engagement office also had previously, about 6 to 8 months previously or so, it was late last year, introduced us to our industry expert and business mentor, Dr. Jeff Spisner, who is supporting our efforts both in the laboratory and on the pathway to commercialization. Jeff is a local biotechnology entrepreneurial leader, launched multiple biotech startups, successful multiple biotech companies in the Columbus area. He has lots of experience bringing biotechnology products to the market and experience with FDA regulatory affairs also. So we have learned over the last few months that indeed the pathway to driving an idea concept towards commercialization is a true team effort with multiple individuals with unique expertise and experience that range from technical, legal, commercialization, clinical diagnostic testing, and industry experience, all coming together around the table with the idea concept at the center, both inside and outside of OSU. So this is similar to the Buckeye football team scoring a touchdown. In order for that to happen, all the members of the offense have to block all individual members of the defense to prevent them from tackling the ball carrier so the ball carrier can cross the goal line. And when all of these individuals, our partners, come together around our innovation, we are beginning to feel a sense of synergy that we think is necessary to hopefully drive our technology from the drawing board, as Patrick introduced you to, through the idea concept phase, where we are now, all the way to market. And as you can tell by a couple of football references, indeed, I'm going to miss the Buckeyes playing this fall. But I wanted to say thank you to all of our partners at the OSU Office of Research, especially Dr. Jan Weisenberger and Dr. Morley Stone. I'd like to thank Janelle, Kate, and Art at the OSU Corporate Engagement Office, Moo Yang and Wayne Embry at Revone Ventures, and Drs. Jean-Michel Balada-Lassatte, Nicholas Wangschwart, and Jeff Spisner. And lastly, thank you to Carolyn Crucifoli and Tanya Matthew at the CCTS for the opportunity to present on this exciting platform today. Indeed, there are so many institutes, as you see here, that have been involved in supporting our efforts, and we are all very grateful for all of them. So the next, we're going to highlight just a few of them. You will hear from Dr. Jan Weisenberger from the OSU Office of Research. You'll hear from Dr. Art Gouret from the Corporate Engagement Office, And finally, from Moo Yang from Revone Ventures to talk about the support that their respective institutes offer to OSU and nationwide children's hospital investigators. With that, it's a privilege to introduce Dr. Jan Weisenberger from the OSU Office of Research. Thank you all.
Thank you, Chris. It is truly a pleasure to be here today, and it was a delight to hear the discussion of the team effort that all pulled together to move the project of Carlos and his team forward. I want to say something right up front about the fact that I am always impressed by the breadth and expertise in the research enterprise at Ohio State, but I have never been more proud and more grateful to our entire research enterprise over the way that they have pulled together and they have brought their expertise, their innovative spirit, and their generosity, their collaborative effort towards addressing the pandemic, also toward helping us to reopen the research effort at Ohio State across the entire university. Next slide, please. So our office provides three main functions for researchers at the institution. One is compliance. We provide through various offices like the Responsible Research Practices Office, the Office of Research Integrity, and the Office of Research Secure Research. We provide tools that help keep our faculty, staff, and students, as we say, on the right side of the law. We want to make sure that the research is done in compliance with all the regulations. Secondly, we oversee operation of research across the entire university. We help to apply for, execute, and manage research grants through our Office of Sponsored Programs. And then finally, our office serves a role in strategic direction and strategic investment in interdisciplinary research that aligns with both short and long-term university strategic priorities. Through that, we have a number of centers and institutes that report up through this office. Next slide, please. So what have we been up to lately? Well, first, none of us are actually working on any of the things we thought we'd be working on this summer. But we've spent a lot of time reopening our research enterprise, which was yet another team effort where our office worked together with facilities operations and development and with university purchasing to make sure that as we staged the reopening of research, we took the recommendations of a faculty committee about the risk associated with different research activities, turned that into a staged reopening that required close coordination in opening buildings, making sure signage was in place, with purchasing to make sure PPE was available. And we are now in the fifth and final stage of our research enterprise reopening. The second thing we did, you heard a little bit about this, was we ran a COVID-19 seed funding program. I will talk about that in a second. I also want to highlight two current opportunities for those of you who are here today to know a little bit about other things that you can get involved in under the auspices of the Office of Research. Next slide, please. The COVID-19 seed funding program was a real fire drill. We received 259 proposals in 2.5 weeks from 642 PIs and co-investigators from 12 colleges at Ohio State across 4 of our campuses. After 2.5 weeks, we stopped, mostly because we ran out of money. We awarded about $777,000 in seed funding. And we had many, many tired reviewers at the end of that 2.5 week period because we had to review all 259 of those proposals. Next slide, please. The proposals were astonishing in their breadth. And so this is just a partial word cloud around some of the topics that we received proposals around, such as vaccine development, diagnostic testing of the sort you heard about from Carlos and his team today, RNA modification of the virus, development of new reagents, and then a lot of things on the social and behavioral sciences side. What are the decision behaviors that people engage in risky behaviors during the time of a pandemic? How do you best message to get good compliance with things like mask wearing? What is the overall economic impact of the shutdown on our economy, both regionally and nationally? And then finally, what are the effects of a pandemic on individuals with fragile mental health conditions? So we were very, very gratified by the range of responses, and we are very, very gratified by the generosity of people putting their talents to work to address an incredibly serious pandemic. Next slide, please. I want to talk very quickly about two current things that we're doing. One is growing the research enterprise through our program Growing Research Opportunities. This is the opportunity to put in a proposal to host workshops of one of two kinds. The first kind are ideation workshops, where a group of faculty members are already in a team. They have a big idea and they want to pursue a large external funding opportunity within a 12 to 24 month time frame. And this is a workshop that allows them to solidify their ideas, form their capture strategy, make sure they have all the right partners. The other type of workshop is a little more imaginative. It is a futures workshop. What are the next big leap research questions that are coming along? And who are the people who are interested in coming together to form teams and come up with specific ideas around those big research opportunities? If you would like more information about either of these, we have several underway at the moment. There's a link down here at the bottom of this slide that you can check out. And then the last thing I'd like to mention very quickly is another opportunity that is open right at this minute, the Racial Justice Seat Grant Program. The goal of this program is to address historic and contemporary effects of racism and racial disparities found in all aspects of American society. We are expecting proposals. They need to be interdisciplinary, innovative, and translational. The call for this is open right now. The first round of proposals is due September 15th. If you'd like more information about this one, there's a link here in the slides as well for you. So that's what I have for you today. I would like next to introduce one of our great partners across this institution, our Technology Commercialization Office. And today, here from TCO is Art Gouray. Art.
Thanks, Jan, and good afternoon, everyone, and happy to be here. A consistent theme we've heard from Carlos and his team is the importance of partnership. And that's where we come in our office. We help put together all the partnerships that would lead to successful commercialization, how we understand the early stage technology and what processes we have in place to move it along the commercialization pipeline. With that in mind, I want to point out that we focus a lot on what I would refer to as platform technologies. And Carlos and his team is a great example of a platform technology. Can we go to the next slide, please? So what I'm showing here is on the so-called x-axis is your classical commercialization pipeline. You have your early stage fundamental science, and we are really great here at AOSU in carrying out some great fundamental science and great R&D and funding are so associated with that. I used in any commercialization process, we have a metric that we follow through to so-called gauge the readiness as we move along, and one that we all use and familiar with is a technology readiness level, a TRL. Whether we use one that DoD or NIH, they all have their NASA, they all have their own technology readiness level, also NSF to some level. And so we get these early stage technologies, and what we like to do is, can we move these early stage technologies to where we get to the startups and licensing, and which are things that we carry out in our office. So we have to be able to get there, and a key to get there, as Carlos and his team, is to get into what I refer to this proof of concept. And then from proof of concept, you move to your advanced development. So what are the partnerships that we need to move successfully along that commercialization pipeline? Is that we have to understand what it takes to get a an integrated technology set that's ready. And that's where we come in with our offices and our experiences. We focus on a total solution that we're going to provide to the customer. And as one could imagine, not everyone has all the core competencies that need to come in to form that systems integration, that total solution. So what we look at now is what are the partnerships we have to bring in place? What are all the key value chain partnerships? And that's the key here. We worked with our I refer to them as our co-development partners. We work, of course, with the colleges, with all the colleges, and to do that, we get great support from folks like Revone. We work a lot closely with our research centers at the university, the CDME, the ESL, and see what all that kind of partnership that we need to bring in so that we could move it along. And we in our office, like myself, I have over 30 years commercializing technology. So we have a line of sight to the customer, how the customer is going to use that product, and that when we get into this advanced development, we want to make sure that we carry out experiments that simulate typical customer usage. Important now is also we want to align all the technologies that we're doing with these product platforms that are out there in terms of the industry partners, the folks we're going to be partnering with. And lastly, as Scott mentioned, in our office, we also want to be, we also do provide entrepreneurial education. Whether it's for faculty or to students through our Keenan Center. And so part of these best practices for commercialization and learning, we deploy some of these learning as we go along. So could we go to the next slide, please? So the next few slides I have is nothing more than a little more details on what I had in terms of this partnership. Many of you might recognize this slide. It's something that NIH and NSF use. And again, on the x-axis is what I had in the previous slide. Your typical commercialization process, and they refer to that proof of concept, that translational research, as your valley of death. You have to really do a lot of investment in here, not only money, but investment in the technology, has the right set of what I refer to co-development partners to make sure that we could exit. I prefer to call it our, as I did in my previous slide, our valley of opportunity. So more importantly is that we need to make sure we understand what is, say, a TRL, technology readiness 3 through 6, because if we're successful there, that gets us through this valley. Now, what is it we have in our office that could that really helps get us through this valley of death is that we have something up here. We have our accelerator award, and that's something that's half funded by the state, half from the state by the university, and it's a proof of concept. So we work very, very closely with the faculty, make sure we assess the technology, carefully define where we are in technology readiness, and work with them to put together a really good accelerator award, and then work with them during the award and the post-award to move the technology along. So you have that OSU, and then we have NSF, as Carlos and his team has. I work with them to go after an NSF, and again, that NSF is called a PFI, and its key word there is partnership for innovation. Work with the team to put together right partnerships, and then after that could lead to the SBIR, STTR. In this case, this is the point where you get into startups, because for SBIR, STTR, the startup is the PI for these types of activities. So we have a good funding ecosystem mechanism to help move these technologies through this valley of death and to be successful. If you go to the next slide, again, it's the same theme as I mentioned. It's our commercialization process I have, but what I have here broken down is more some of the details that we go through. And actually, all of these items that I have here is what I spent with Carlos and his team to make sure that we have a good successful application. At the end of the day, at the university, we want to, what I'm showing here, we want to be at the stage where we could successfully transfer the technology, transfer it to design manufacturing. Granted, I spent 30 something years in that downstream design manufacturing, all the go to market, but we want to make sure that we do a good job on that upfront technology engine here for this product development or technology commercialization process, so we could translate. So we want to get it to a good level of technology readiness. Now, I want to point out that when I work with a team, like Carlos said in his team, is that we focus on a few key items that goes into any good partnership proposal. We work in our team, we do a market analysis, we do an IP landscape analysis, we do a competitive analysis, and then also now a key part of any of these proposals is what is referred to as a commercialization plan. We do have the expertise to help with all of these aspects that goes into a proposal. My last slide is I just want to point out that we have some great success stories here that in our office, we've worked with faculty. If I point into this failure, into the TRL3, where we identify failure modes, that's my definition of technology readiness. We've been extremely successful at NSF PFI. I would say is that we're batting 100%. Every proposal we work on and got submitted to NSF for the PFI, the TT, and the RT, we've been successful. We've been very successful at the SBIR, STTR, particularly with AFRL, and both phase 1 and phase 2. And so what this last slide is showing is the partnership and the funding mechanism that we have in place to move the technology along the commercialization pipeline. So we're here to partner with you, to understand your early stage technologies and put things in place to make sure that we could successfully move it along a credible commercialization path. And thank you. And with that, I would like to introduce to you Moo. Moo, as I mentioned, has been a great partner for us in Revone, and she and I work closely on a lot of these proposals. And I'll hand it over to Moo.
Thank you so much, Ari. It's such a pleasure to be here. So my name is Moo Yang, Moo from Revone Ventures. So I'm a director at Revone, being an advisor for life science companies. So just a little bit kind of talking about our vision and also what is in our blood and DNA. We're here really to help entrepreneurs build great companies and also help the corporates to innovate. So Columbus is actually one of the areas to have such a huge privilege to having multiple corporates located, headquartered here. So that's what we're here to do. So Revone Ventures, we actually call ourselves as an investor startup studio. So what does that mean specifically is that we bring capital as an investor and also strategic services to the table. So on the service side, I would love to draw your attention to kind of the team connections and space we have. So we do help a lot of prolific innovators from research institutions to identify their talent team needs when they're ready to actually spin out a technology to build a startups. And also me and myself as an advisor, along with other very experienced team that we have at Revone, can help you advising along the way. And then as Patrick and Chris talked about earlier, we have learning labs, and customer learning lab being one of them. And we have many other different kinds of learning labs kind of help you work you through a commercialization pathway. And then quickly, I do want to give a quick shout out of our internship program that we just announced our continuation for the fourth cohort. So this internship program is in partnership with the Ohio Development Service Agency that we will be able to provide not only the administrative program support, but also financial support by offsetting up to 50% of the salaries for the interns. So if you're interested, please check out revoneventures.com. And then obviously we have connections, that we have the corporate partners in our backyard that we are ready and willing to help you when you're ready to make those introductions, to either have a pilot study with them or even get them as your initial customers. And we see that a lot of times. And then obviously for the investment side of things, we co-invest with a lot of regional investors. So CCTAG, that Jennifer has briefly mentioned, that we do a lot of co-investment with them as well in Cincinnati. Now space, if you have not already, we actually manage a pretty sizable space on the Kinnear Road. Is on the west side of a campus. And then we host about 50 plus startups there. And those space becomes such an instrumental foundation for a lot of startups when they first start, so that they can build on top of what they have and then have the access to wet and dry lab space that we have. So next slide, please. So in case you are wondering, what does Revone do specifically? So just give you some flavor of kind of the 2019 startup impact that we were able to achieve. So in 2019, there are 33 companies that were funded through a variety of funding mechanisms we have. So as a result, we actually deployed about 14.3 million in capital. And then we're really proud that 42% of the company that we funded have diverse leadership. That means anyone on the management team level and the research funding team that we have working with OSU and other partners in the region. And then as a result, there are over 1,000 jobs created and retained. That's always kind of the goal, that why we're doing what we do here in Central Ohio. And then those startups that we supported, they generated revenue in a range of $148.8 million. And those companies, because they need to attract additional capital, they were able to actually attract additional $170 million. And then on the sum of the good exits that we have, resulted over 200 million in 2019, which is always nice for some of the funding vehicles that we have. So in total, the total impact in 2019 alone, we were able to achieve roughly about 530.4 million in that. So we're really proud, and that's why we're here to help you, support you. That's what we do. So next slide, please. So this is my last slide. I really want to point out Ohio State University is our innovation partner. So we're really here to help build great startups. So in the past, we actually have helped brilliant faculty members like yourself from the audience from 15 colleges and all other units around the university to drive those innovation by filing innovation disclosures each year. So roughly it's over 400 invention disclosures have been filed, and this has translated into nearly 300 active licenses and technologies invented at Ohio State that are impacting lives around the world. So one of the leading testament is today's technology with the DNA technology team. So with that being said, it's a great pleasure to be here. And next, I would love to introduce Dr. Bruce Weinberg, who is the program director at CCTS and also professor of economics at OSU. And we will be officially start the Q&A session. And I highly encourage you to put your questions in the Q&A box. Bruce?
Thank you very much, Moo. And thanks to everyone for joining us. Just in the interest of time, so that we can wrap up on time, I think I'm going to turn things over to Becky Jackson for some concluding remarks. And one of the things that I did want to do as part of the concluding remarks is to wish Becky a happy birthday. So I'm going to turn it over to CCTS Director Becky Jackson.
Thank you, Bruce, for those kind words. My name's Dr. Rebecca Jackson. I'm the director of the Ohio State University Center for Clinical and Translational Science. And I want to thank all of our presenters today that really helped to illuminate some of the messages and some of the aspects of the journey from discovery all the way to dissemination. First of all, our Inventor team, led by Dr. Carlos Castro, and importantly as well, those that represent some of the innovation resources available here at The Ohio State University and in Central Ohio. This includes Dr. Jan Weisenberger from the Ohio State Office of Research, Dr. Art Gouray from the Ohio State Corporate Engagement Office and the Faculty Alliances for Engineering, and Moo Yang from Revone Ventures. At the Center for Clinical and Translational Science, our mission is to enhance the translation of discovery to new treatments and prevention strategies to improve human health. And we do this from really three special components, from the development of actually new teams to stimulate passion, to identify pressing scientific questions that you heard today in response to the COVID-19 pandemic, to establish diverse collaborative team, and then to bring in other individuals that actually help to support the journey. We then demonstrate innovative and potential solutions to ensure both a culture of safety as well as to enhance the rigor and reproducibility, and then share this knowledge as we did through a program like today of how one goes through this process of innovation, commercialization, and entrepreneurship. The key component and what we consider to be the special sauce of translational science is in fact team science, bringing together people from multiple disciplines and expertise in order to be able to solve a problem. And I think as we've heard our team talk about today, it really does take a village to be able to address the COVID-19 pandemic and so many of the other issues that are facing us that will ultimately enhance human health. I invite you to come back again next month for the next of our workshop series to hear Dr. Parina Guma, Andrew Bowman, and Matthew Exline presenting their innovations around a breath test for COVID-19. So that's September 22nd from 12 noon to 1 p.m. right here on your favorite e-channel. We also want to make you aware of a new opportunity that'll be available next month as well. And this will actually be an SBIR, STTR Biosciences company showcase, highlighting the work of Dr. Jessica Winters and bringing us some real insight about some new program from the lead program officer in the SBIR Development Center at the National Cancer Institute. So we hope that we'll see all of you at that event tomorrow. The SBIR and this STTR Biosciences Company Showcase is co-presented along with the CCTS and the Office of Research with G2G Consulting, BioOhio, and Revone Ventures. Again, thank you so much for your attendance today. Please feel free to contact any of us with questions, the speakers, or any of the other members of our invention support team, and we'd be happy to help you. Take care. Have a great day. Be safe. Wear your masks.